So molasses is mother liquor that is obtained during the crystallization of sugar, which is prepared from sugarcane juice. Okay. Then molasses contains only 30 to 40% of sugar. Okay. So that is mother liquor, brown color syrup molasses. So percentage of sugar present in molasses is only 30 to 40%. Okay. So molasses contains 30 to 40% sugar. Okay. So this is molasses is 30 to 40% concentrated sugar. Okay. So this 30 to 40% sugar containing molasses first gets diluted with the addition of water. Okay. So next step is molasses gets diluted to 10% concentrated solution with the addition of water to this. Okay. So molasses diluted with the addition of water to 10% concentrated sugar solution. Okay. So with the addition of water it gets diluted and now concentration is 10% concentrated. Okay. After that we have to add H2SO4, few drops of H2SO4 to 10% diluted sugar solution. So here purpose of addition of H2SO4 is to maintain the pH of the solution at 4. Okay. So 10% sugar solution is there. Okay. So molasses on dilution gives 10% sugar solution. So to this we have to add H2SO4 solution. Okay. So why H2SO4 solution is added to this? So added to 10% solution to maintain its pH at 4. Okay. Then to this next step is yeast, addition of yeast. Okay. And yeast is added to this solution. Okay. So what is the purpose of yeast here? Yeast is microorganism. It produces enzymes. Okay. So here yeast produces enzymes like invertase and zymase. Okay. So here after the addition of sulfuric acid to maintain pH at 4 and after the addition of sulfuric acid we have to add the yeast. Yeast that is to produce the microorganism, to produce the enzymes. Okay, yeast is microorganism and it produces enzymes. Okay, then this 10% diluted solution that is cooled to 30 degrees centigrade and kept for 2 to 3 days. Okay. And one more thing is after the addition of yeast, we may add ammonium sulfate or ammonium phosphate. Okay. So to yeast, to this solution after the addition of yeast, we may use ammonium sulfate or ammonium phosphate. Okay. Ammonium sulfate or ammonium phosphate are added to this solution. Here purpose of ammonium sulfate and ammonium phosphate, that is food for yeast. Okay. So these chemicals, they are used as food for yeast to produce enzymes fastly. Okay. Then after that 10% dilute solution that is cooled to 30 degrees centigrade. So 10% solution gets cooled and temperature is 30 degrees centigrade. and it is kept for 2 to 3 days. Okay? Kept for 2 to 3 days. Okay? So during this period, so yeast produces zymase invertase enzymes. Okay? So in this time of period, yeast produces enzymes like invertase and zymase. Okay? Then, so during this period, sucrose slowly converts into ethyl alcohol. Okay? And this is two-step process. First step is sucrose, in presence of invertase enzyme. Okay? It converts into glucose and fructose. 1 mole of glucose and 1 mole of fructose. Okay? This is sucrose, which is present in molasses. Okay? And in presence of invertase enzyme, it forms 1 mole of glucose and 1 mole of fructose. 1 mole of glucose and 1 mole of fructose. Okay? And this reaction is also called as inversion of sugars. Okay, where invertase enzyme is present. And in next step, either glucose or fructose in presence of zymase enzyme converts into ethyl alcohol. Same as in starch, preparation from starch. There also glucose, only maltose on hydrolysis forms 2 moles of glucose only. But here we get glucose and also fructose. That's why glucose or fructose. This is glucose or fructose in presence of zymase enzyme. So enzyme here is zymase enzyme. We get ethyl alcohol along with the CO2. And here ethyl alcohol which is obtained in this reaction, this is called wash or wort. Okay? 6 to 10% ethyl alcohol is obtained here. This is called wash or wort. Okay? And it undergoes fractional distillation. Okay? And we get 95% alcohol. This is called rectified spirit. Okay? Next, rectified spirit undergoes further distillation in presence of calcium oxide. Okay? It undergoes distillation in presence of calcium oxide. And here we get 100% alcohol. This is called absolute alcohol. Absolute alcohol. Okay? If methyl alcohol is added to absolute alcohol and it is unfit for drinking purpose. Okay? So, if we add methyl alcohol, CS3OH and 100% absolute alcohol, we get denatured spirit. Denatured spirit. So, denatured spirit, this is unfit for drinking. Why? Because methyl alcohol is poisonous in nature. So, that's why it is unfit for drinking. Okay? Then 95.5% ethyl alcohol and 4.5% water mixer. This is called azeotropic mixer or constant boiling mixer. Okay. So all this is about the preparation of alcohol. So during fermentation process we get only 6 to 10% ethyl alcohol, which is called wash or wort, and on distillation it forms rectified spirit, and further distillation it forms absolute alcohol. With the addition of poisonous methyl alcohol it is unfit for drinking and it is called as denatured spirit. And constant boiling mixture, which is also called as azeotropic mixture, is 95.5% ethyl alcohol and 4.5% of water.